Dearest little KCET aspirants, I am here today to teach microbes and human welfare in one stroke. So this is a Masterstroke, my dear children, and I'll tell you, this is one of my favorite chapter from class 12th. You might ask why there are so many names, but you know what? If you remember those names, this chapter can give you marks like this because there is nothing extra that could be asked around it. So you get it? So make sure you are remembering the names. You will be remembering it if you just go through this chapter peacefully. You would be able to remember it if you associate with things and learn. If it's a fun learning, you will definitely remember it, and I want one thing from you, that is your NCERT and a pen, my dear children. So whenever I say that this area could also give you questions, but you do not usually prioritize this, so you get to know, okay, I have to prioritize this also. And apart from that, when you have your NCERT in front of you, when I am teaching you, you get to know where I am teaching from. It's like you can relate and learn, and that basically the pictorial memory and everything, when you know that this particular thing is in this particular page, you might remember things more better. So are we ready? Yes, we are ready, I hope so. Well, there is no other way to learn that. So the first thing is microbes in household products. So in house, what all you use? You have milk, you have cheese, and sometimes we make dosa, idli and things like that, right? So for all these things, you need certain microbes, my dear children, which are like our best friends for daily use. What are those? So first thing is lactic acid bacteria. Okay, lactic acid bacteria, as you can understand, they might be the ones that might be producing lactic acid in the food. Okay, and you know what? When these bacteria are added into the milk, it converts the milk into something that is known as curd, my dear children. So LAB, that is lactic acid bacteria, produces acid. This acid basically coagulates and partially digests the milk protein. In the milk, there is a protein whose name is casein. This casein, when it comes in contact with the acids released by the bacteria, it is going to have a disrupted shape and structure and everything. So it is going to coagulate, it is going to partially digest it, which is why it will now form lumps sort of thing, which is known as curds. Now also one more thing, this particular bacteria is responsible for increasing vitamin B12 content in the curd. That is why we are asked to take curd, because it has vitamin B12. You know, some people have injections also for vitamin B12 and not at very good places, so you feel the pain for a bit more time. So obviously, if you do not want those injections, definitely have some good amount of curd if you're not allergic to it. Yeah. Now LAB in the stomach, it prevents harmful microbes as well. So lactic acid bacteria are really important. I hope you will never forget it. So it is basically added to milk, and in the milk they are going to produce some acids, and these acids are going to partially digest the milk protein, that is casein. And when you do that, basically structure and everything is changed, which is why everything will coagulate and form lump sort of structures, and together I'll say now the milk has turned into what? A curd, my dear children. Well, while they are doing so, they are also increasing the content of vitamin B12 in the curd, which is why curd is really important. Got it? Works? Now let's go to the next thing, that is, let's make some dosa and idli. So over here, my dear children, bacteria, there are certain bacteria which is going to ferment the dough. Okay, when fermentation process happens, you know what happens? When fermentation process happens, there is a production of CO2 and there is a production of alcohol. Okay, so it's not like the alcohol which makes you go like this. No, not that much amount of alcohol, very little amount of alcohol. So certain bacteria is added, and basically they produce what? CO2, and also this CO2 is the one that will be puffing up the dough. Okay, the CO2 which is released, my dear children, it puffs up the dough. And apart from that, if you might have heard about yeast, my dear children, Saccharomyces cerevisiae. Okay, now it is known as baker's yeast because this yeast we are going to use in bread making. In bread making, it is known as baker's yeast. If you want to make something else, it will have some other name. So baker's yeast, Saccharomyces cerevisiae, it is used in bread making. Same thing. Yeast also will undergo fermentation. While it undergoes fermentation, my dear children, it is going to produce CO2. And this CO2 is something that is basically going to give little holes and make it spongy and puffy. What is it? Bread. Okay, so please remember the bread thing, that is baker's yeast, Saccharomyces cerevisiae. Now, traditional drinks and foods, that is also there. So you might have heard about toddy. Okay, in Kerala and all, if you go, there are toddy shops here and there. Okay, so what is it actually? You take the palm sap, okay, the sap of the palm, and then ferment it. So when you do the fermentation, I told you get two things. One is CO2, another one is alcohol. So little amount of alcohol content basically increases over here, my dear children. So one such thing is that now fermented foods are there, such as fish, soybean, bamboo shoots, and everything can be fermented as well. Okay, so that's all. Okay, now there's a question, I think it's not here. So for malted cereals and all, they are also fermented and they are being used. But what is that organism used to ferment all this thing? It is Saccharomyces cerevisiae again, my dear children. I think it will come again, but as of now you have to remember it. Now, the fourth thing is cheese production. Now cheese, no, there are two examples given in a book. One is Swiss cheese and the other one is Roquefort cheese. Now Swiss cheese, no, is the one that Jerry used to take away from, it used to come off of its bill and used to carry this particular cheese. Okay, and try to escape Tom. So basically this cheese, my dear children, is Swiss cheese, and as you can see, it has big holes. It has big holes. And why is there big holes? Because the bacteria which is in there, its name is Propionibacterium shermani. Okay, what does it do? It releases CO2, and while it releases CO2, it gives rise to what? The big pores over here. So what is the name of the bacterium? Propionibacterium shermani. That is the name. So it is in Hindi, shermani means it is shy. So because it is releasing CO2, probably it's shy. You can remember like that. I know it's not a very good way to learn, but this is how we learn. Okay. Now Roquefort cheese, if you talk about that, the ripening of Roquefort cheese has been done by a specific fungi, my dear children. So here the fungus plays a major role. They have not mentioned which fungus, so you do not have to remember it. But at least this thing you should know that Roquefort cheese is ripened by using a fungus. So over here, curd formation, you have lactic acid bacteria, that is lactobacillus, that is the name of the bacteria. It is going to release acids, is going to convert milk to curd and also increase the vitamin B12 amount. Okay, now if you're talking about bread making, in bread making you need baker's yeast, which is also known as Saccharomyces cerevisiae. Okay, now Saccharomyces cerevisiae would be used for so many other things also. You are going to learn about that. So first thing, it is known as baker's yeast because we are baking our bread with the help of that. Now there is toddy, my dear children. Toddy is basically nothing but fermented plant sap. As I told you, whenever fermentation happens, CO2 is being released and also alcohol is being formed. So toddy definitely has content of alcohol in a very much amount. Okay. Now if you talk about cheese production, you have two cheese mentioned in your NCERT, that is Swiss cheese and Roquefort cheese. So Swiss cheese, my dear children, it has big holes, and why is it? Because the bacteria which is in there, okay, that is Propionibacterium shermani, it is releasing CO2, and while it releases, so it creates pores over there. Now talking about Roquefort cheese, it is ripened by using a fungus, not a bacteria, it's a fungus. Yeast, if you know, yeast is also a fungi. Okay, yeast is also a fungi, only unicellular fungi. Rest all of them are multicellular, but the yeast is unicellular. I hope this is done. So household products is over. Going ahead, if we talk about industrial products, my dear children, so again microbes are used in industries also. It is used to synthesize valuable products like beverages and antibiotics. It is grown in large fermenters, my dear children, because they are needed in large amount. So we will see first of all, in beverage production, what is the use? So now the Saccharomyces cerevisiae is known as brewer's yeast. Why? Because we are going to make beverages out of it. So it is used to ferment malted cereals and fruit juices. I told you it is used to ferment malted cereals and fruit juices. This is a question also. Okay, what is that? Something that is used for fermentation of malted cereals and fruit juices? It is brewer's yeast, that is Saccharomyces cerevisiae again. And while it does so, as I told, fermentation means obviously you have to produce what? Ethanol. You have to produce ethanol. So over here, ethanol is going to be produced. Now, fermentation pathway, that is converts, basically how does fermentation works? Whatever sugar is there, my dear children, it is broken down to ethanol and CO2. That is given. Now the types of alcoholic drinks. So there is beer, there is wine, there is whiskey, there is brandy and rum. So two of them you get without distillation, and basically three of them you get with distillation. So what do you mean by without distillation and with distillation? So what happens in distillation is that basically the water content which is there, it is being vaporized and everything. So when the water content is vaporized, my dear children, don't you think what is left back will be a concentrated form of alcohol? Yes, it will be concentrated form of alcohol. So the one that undergoes distillation, it will be having what? It will be having concentrated alcohol. One that does not undergo distillation, that means still little amount of water is in there. It is not too much concentrated. That amount of alcohol will be comparatively lesser. So those are the ones that are produced without distillation. So wine, everyone drinks wine. Okay, beer, not everyone should drink. Okay, so basically, but it also have less content of alcohol in comparison to whiskey, brandy and rum. So they will be having more concentrated alcohol because they are produced with distillation. If you talk about without distillation, it's wine and beer. Again, a very important question. So from this particular part, you have to remember two things. That is, first, there is brewer's yeast, that is Saccharomyces cerevisiae, and it is the one that is used to ferment malted cereals and fruit juices, and it gives ethanol, my dear children. Second thing that you have to remember is the types of alcoholic drinks that are produced. Two of them with distillation, three of them with distillation, that is whiskey, brandy and rum, and two of them without distillation, that is wine and beer. Done? Cool. Now going ahead, we have certain chemicals, certain enzymes, certain bioactive molecules from the microbes that are being produced. Okay, so industrial chemicals, again in the industrial group you can add them. So you have citric acid, acetic acid, butyric acid, lactic acid and ethanol. Okay, so citric acid, my dear children, it is produced by Aspergillus niger. Aspergillus niger. Now remember, this is the only fungus over here. Rest three of them basically, they are bacteria. Okay, there are bacteria. I am not considering ethanol over here, so I just put ethanol. But let's see, acid. If I talk about acid, citric acid is the only thing that is produced by a fungus, and that is Aspergillus niger. Talking about acetic acid, the name is very much common. It is produced by Acetobacter aceti. So Acetobacter aceti produces acetic acid. It has something in common, right? That is a bacterium. Now butyric acid is produced by Clostridium botulicum. Okay, what is it? Clostridium botulicum. This is again a bacteria. Lactic acid, you know, it is produced by Lactobacillus or lactic acid bacteria, my dear children. It's again a bacteria. Talking about ethanol, we studied it. It is produced by Saccharomyces cerevisiae, that is yeast, again a fungus. Okay, so citric acid by Aspergillus niger, a fungus. Acetic acid by Acetobacter aceti. Okay, butyric acid by Clostridium botulicum, again a bacterium, and then ethanol if you want, Saccharomyces cerevisiae, which is a yeast. Now enzymes that you get from the microbes, you know, there are enzymes such as lipases. You know, lipases are going to act on lipids, my dear children, are going to act on lipids. For example, fat is a form of lipid, right? So basically I have got some oil stain on my dress, so I would be using detergents which has lipases in them, so that these fat can be broken down. So lipases are used in detergents to remove oily stains. So this question can be coming. Okay, now to clarify bottled was say fruit juices and everything, okay, to make the break down the lumps and everything, we have pectinases and also proteases. It is proteases are going to break down the proteins over there. Okay, pectinases are going to break down the pectin, which is a part of the plant cell. Okay, cell wall. Got it. Now you have, okay, so this is for something else. So these are the enzymes that are being produced by microbes. Or shall we include this also? Okay, let's include this also. Fine. So lipases is one such thing, and then pectinases and proteases is one such thing. Now this is very much important. Streptokinase. Now streptokinase, whenever term A is come, that means it's a bacteria. Sorry, it's an enzyme. Now streptokinase is produced by what? Streptococcus, which is a bacteria, my dear children. And you know what it does? It bursts down, okay, it bursts down the clots which are there. Obviously, if we have clot, we will be dying. Okay, we do not want clot in our blood vessels. So it is going to act like a clot buster, my dear children. What is it? It is streptokinase produced by Streptococcus. Okay, and it is used for heart attack patients and everyone. So this is one very important thing. Now there are certain bioactive molecules which are being produced by basically some microbes. So they are not enzymes, they are just molecules which are being produced. So you have cyclosporin A. Cyclosporin A, it is produced by Trichoderma polysporum. Trichoderma polysporum is a fungi. Okay, what is it producing? Cyclosporin A. When is it given? For example, I had a organ transplantation surgery. Now my body thinks this is something foreign that I have put up over here. My body does not feel good about it. Who is this thing inside my body? I should be basically breaking it down. So I do not want to do that. I want my body to understand that it's now going to be my own part. So basically at this point of time, since my body is not that understanding, what do? We give immunosuppressants so that my immune system is bit suppressed. It does not react to the foreign particle which is there, that is the organ which is there. Okay, so cyclosporin A, which is produced by Trichoderma polysporum, is going to act like an immunosuppressant. Okay, now the next thing is statin. Statin is produced by Monascus purpureus, my dear children. What is it? Monascus purpureus. And statin stabilizes, I remember like that, statin stabilizes the blood cholesterol level. So it lowers cholesterol by inhibiting its symptom. It lowers cholesterol by basically inhibiting its not symptom, its synthesis. Its synthesis is being inhibited because it goes and basically competes with the enzyme and everything out there which is responsible for synthesis of cholesterol. That's how it works. So we are not going that deep, but at least you should be knowing it. Okay, so I'll actually put these three things together because it's totally different from what we are learning over here. So that's how I usually learn it. Okay, that's how I will recommend you to. Okay, so let's sum up. So first thing, there are certain acids which are being used, my dear children, at our household works and also for a lot of other things. Okay, now first thing is citric acid. Citric acid is basically produced from Aspergillus niger, which is a fungus. Okay, acetic acid, Acetobacter aceti, which is basically bacterium. Okay, butyric acid by Clostridium butylicum, which is again a bacterium, and ethanol by Saccharomyces cerevisiae, which is yeast, my dear children, which is a fungus. Okay, apart from that, there are certain microbes which produces lipases. Lipases are there for removing lipids or fats. So for removing oily stains, I can use lipases. Now I can use pectinase and also protease for in the bottled fruit juices, my dear children. It will be breaking down everything and making the juice really good. Okay, now we have three things that you have to remember. Streptococcus is a bacteria that produces streptokinase, and streptokinase is going to act like a clot buster for heart attack patients, for person who have undergone myocardial infarction and all. They can take streptokinase. Now you have cyclosporin A, which is produced by a fungus, whose name is what? Trichoderma polysporum. Okay, and what does it do? It is acting as an immunosuppressant. Now there is statin being produced by Monascus purpurus, my dear children, and it stabilizes or lowers the blood cholesterol level by inhibiting its synthesis. It does not allow to synthesize. Got it? So these are the things that we studied as of now. Now you might feel, how is ma'am able to say all this thing, or how ma'am thinks that we are also like her only. Are, I am also like you actually. Okay, it's not like I learned it in a day. No, I taught like several times. That's how I learned it. It's not a one-day process which is there, my dear children. So I'll recommend you, while you're talking to someone, with your parent and all, you just give them the paper and ask them, is it correct? Is it correct? Just talk about it. And don't think that I have to learn this, I have to remember this, then only you can. It's not like that. Do not pressurize your brain. Go with the flow. Go with the flow. If at your home someone has cholesterol, say that you should be taking statin from Monascus purpureus and things like that. That's how you learn. Okay, or randomly call your friend and talk about something out there. Okay, or ask them questions. If they are also learning biology, ask them certain questions. They'll also ask you questions. This might give you panic attack, but I don't want you to do that. Basically, after learning the chapter, only do this. Otherwise, your friend will also might teach you. So this is done. Cool. Now there are antibiotics and the discovery. You know antibiotic, how it was discovered. Just give me a second. It's kind of too hot up here. I don't know why. Okay. Yeah. So antibiotic and its discovery. So it is very much interesting story. I will tell you. First thing, what do you mean by antibiotics? Antibiotics means against life. It kills or inhibits the microbes. Against life as in whatever organism that has a life, we want to kill it. Okay. So it is beneficial to humans and it is produced by microbes and is used to treat bacterial infection. Antibiotics against bacteria. So first antibiotic was discovered by Alexander Fleming in the year 1928. How? So basically, Mr. Alexander was working on a bacteria that is Staphylococci. Okay, what is it? Staphylococci. So Staphylococci bacteria, my dear children, was growing. So every day what he used to do is he used to wash the petri dish and then go. But one day what happened was, this petri dish was not washed. So Staphylococci bacteria was like, today is my day. I am going to divide myself and I am going to have a wonderful life up here. But what happened is that since it was not washed, a fungi basically grew over here, my dear children. Fungi grew over here and its name was Penicillium notatum. Penicillium notatum. That is no tantrum, you can remember like that. Okay, so Penicillium notatum, my dear children, it was being produced. Okay, so what happened is that this Penicillium notatum were releasing something which was not allowing the Staphylococci to grow, my dear children. So they instantly became sad. What was this something which was being produced? It was penicillin. It was what? Penicillin. The spelling is correct or what? Penicillin, I always forget it. Yes, it is correct. Yeah. So penicillin was being produced. Now, Alexander came the next day. He saw that, oh shit, first thing, I didn't basically wash the petri dish over there. Okay, so the next day Alexander came in, and basically what did he find out was the petri dish is not washed. Oh, what did I do? He was so worried. And then he found that if I didn't wash it, it should have grown. The staphylococci which is there, it should have grown. Why is it not growing? So he went ahead and saw that there is some mold, fungal mold over here. And then he came to know that is Penicillium what notatum, my dear children. And probably this mold might be releasing something which does not allow this to grow. So isn't it acting as an antibiotic? It is not allowing the bacteria to grow. So this thing was penicillin, my dear children. So he found that mold Penicillium notatum inhibits staphylococci growth, later developed by Ernst Chain and Howard Florey. They were the people who actually developed it later on. This guy just found out okay, this something like this is happening and things like that. So first antibiotic discovered by Alexander Fleming, but more about it was known by Ernst Chain as well as Howard Florey, my dear children. Now impact and recognition. It was used in World War II to treat wounded soldiers, my dear children. Fleming and Ernst Chain and also Florey won the Nobel Prize in the year 1945. In the year 1945. Okay, there is something more to add to it. Just give me a second. Okay, so I was talking about this thing. Yeah, so basically Alexander Fleming itself, okay, he was a person who founded Penicillium notatum and also penicillin and everything, but the full effective thing about the antibiotic was found later on by Ernst Chain and Howard Florey. So they went ahead and studied about it a lot. Okay, please remember that. So yes, I hope you understood this whole story and I hope you do not forget this. So this is an antibiotic that is produced from a fungus. Okay, and it is named as Staphylococcus. Please do not write and come Streptococcus and things like that. That is totally different. Streptococcus is the one that produces streptokinase. This is Staphylococci. Okay, now going ahead. Again this thing, we studied this thing. Oh, my dear children, KCET aspirants. So I hope you are well aware about the test series and the crash course that we have launched. If you're not aware, then let me tell you. So there's a test series for you, my dear children. It's completely online. It is for ₹1499. And what are you going to get over here is that there will be 35 full syllabus and chapter wise tests. There will be video solution. There will be one shot revision videos and a paper analysis of 5 years. So depending on the paper analysis, only these papers are being made, and these papers are made by the teachers who have so much experience in teaching KCET students, my dear children. So we will be there, okay, to explain you the answers and tell you the answers, but it's curated by big teachers out there, okay, who have a legit experience in teaching this. Now there is Lakshya KCET crash course as well, which is a 21-day course at our campuses. So if you're there in Bangalore, you can come to the campus and you can then go. Okay, otherwise if you want to stay over here, you're not from Bangalore, you want to have a residential atmosphere, you can stay over here. We'll provide you hostel facility, and there will be daily practice tests. There will be one-on-one doubt session. Complete crash course will be there. Now if you want more information regarding that, you can call to this number, or basically what you can do is you can click on the link in the description box. You might get more idea about it. Works? Chalo. Now next part of this chapter is sewage treatment. Very important thing. So sewage treatment, first thing I'll explain you like that, and then we will read out certain things. Okay, so listen to me first. So in sewage treatment, the first treatment is known as primary treatment. What do you call it? Primary treatment, my dear children. Primary treatment. In primary treatment, there are two major things. One is filtration, and the second thing is sedimentation. That means you have the sewage, certain things will be floating around things because they are lightweighted, so those can be removed with the help of filtration. Certain things will be sedimenting down, so those can be removed with the help of sedimentation. Now what are you left behind? There is an effluent that will be left behind, and this will be known as what, children? This is known as primary effluent. It is known as primary effluent, which will be having so much amount of organic matter in it. Cool? So now this primary effluent is being transferred for the second treatment, which is known as secondary treatment. Okay, and what happens in a secondary treatment? There's a large aeration tank, my dear children. There's a large what tank? Aeration tank. And what do you mean by aeration tank? There will be air provided over here, my dear children. Okay, and in aeration tank, what is there? There is our primary effluent. That means we have our organic matter over here. And since air is provided over here, obviously there will be certain aerobic bacteria that needs oxygen to survive, okay, that works on the basis of oxygen only. So there's aerobic bacteria which is there, which is going to act on the organic matter in the presence of oxygen, my dear children. So here comes a very, very important term, that is BOD, biochemical oxygen demand. Biochemical oxygen demand means the amount of oxygen required by the aerobic bacteria to break down organic matter in 1 liter of water. Simple. You see, aerobic bacteria needs oxygen to break the organic matter down. Okay, if the organic matter is way too much, then obviously aerobic bacteria will need more oxygen. So that means biochemical oxygen demand is more. Okay, if organic matter, if my dear children, organic matter is less, that means aerobic bacteria will need less oxygen, that means BOD will also be less. So until what time we will do the secondary treatment? Until the amount of BOD, biochemical oxygen demand, is reduced, until that time we will do the secondary treatment which is there in the aeration tank, my dear children. And it's not like aerobic bacteria does it by itself. No. There will be certain fungal filaments, and a lot of other things which will be clumping up together, and making certain structures which are known as flocks, my dear children. So it's like a mesh-like a structure which is formed of aerobic bacteria as well as the fungal filaments, as well as all the extracellular thing that they would be releasing, extracellular as in outside the cell. So they would be releasing certain things. So all this thing together forms a mesh-like structure which is known as flocks. Okay, and together it will be acting on the organic matter in the presence of oxygen, my dear children, and we will do so until the amount of BOD is reduced. Fine. Now suppose it is reduced very much. Okay, it is reduced very much. That means what? There is no organic matter up over here. That means there is no organic matter up over here. So what are we going to do? We are going to put the same things into something that is known as a settling tank. Now when we put them into settling tank, what will happen? What will happen, my dear children? This flock which is there, it is going to settle over here. This is the flock that is going to settle over here. What are we left with? We are left with secondary effluent. My dear children. This secondary effluent does not have aerobic, organic matter, because it is already been broken down. So this secondary effluent, my dear children, can be put down to river, stream, because it's all clean. Now over here you do not call them flocks anymore. Instead you will have a different name for this sedimented things, which is known as activated sludge. What do you call it? You call it activated sludge. Now what happens? Basically, this activated sludge, my dear children, a little amount of it will be taken, little amount of it is taken and put back in the aeration tank. Why? Because the next set of primary effluent will be coming and you need some aerobic bacteria. So it is going to act like an inoculum. So that there are bacteria. This bacteria will now divide and do the whole process again. Rest of the thing, okay, rest of the activated sludge which is there, it is passed to the next thing. It is passed down to the next tank, which is known as anaerobic sludge digester. It is known as anaerobic sludge digester. As in the sludge is there, the activated sludge which is there, it is put over here, and in anaerobic condition it is going to get digested. So it will, this activated sludge, what is it having, my dear children? It is having aerobic bacteria, it is having fungal filaments and everything. But now the situation is what? There is no oxygen. It is an anaerobic sludge digester. There is no oxygen. And when there is no oxygen, who will be the one that will be the most happy? It will be anaerobes. That is anaerobic bacteria. Until now, aerobic bacteria were doing everything, but now anaerobic bacteria are going to play a major role over here. Okay, so examples, you have methanogens. Methanogens are anaerobic bacteria. They are able to survive in water, in an area where there is no oxygen. So what will happen? These anaerobic bacteria are going to feed on the aerobic bacteria and the fungal filaments and everything and break it down. And while it does so, it is going to produce biogas, my dear children. It is going to produce biogas. And what does biogas include? It includes methane. it includes CO2, it includes H2S, hydrogen sulfide. So methane, CO2, H2S, everything is being produced. So all of them together, not one. I'll tell all of them together as what? Biogas, my dear children. So these biogas can be used in kitchens, it can be used in for electricity purpose and everything. So when you do sewage treatment, you are clearing down the water that is there, and apart from that you are also producing what? Biogas. I hope this is now clear. Okay, so primary treatment involves filtration and sedimentation. Then the primary effluent which is there, which has a lot of organic matter, is put down into aeration tank. Here air is given. In the presence of oxygen, aerobic bacteria will be so happy, it will group up with some fungal filaments and something to form flocks, and they are going to act on the organic matter. It keeps on acting until the level of BOD, biochemical oxygen demand, goes less. That means the level of organic matter, until it goes less, it keeps on acting. And now what are we left with? We are left with clear water. So this is being transferred to a settling tank with the activated sludge. The flocks and everything will be settling down. You will call this activated sludge. What is left behind is put to river and stream and everything, because the water is all clean. Now what do? You take a little amount of activated sludge and you put it back over here, because it acts like an inoculum. It helps in the growth of bacteria, which will do the treatment over here. Okay. Now the rest of the amount of activated sludge is put down into anaerobic sludge digester, where the anaerobic bacteria will be truly happy, because they are going to feed on the aerobic bacteria as well as the fungal filaments. And while they do so, since it is anaerobic condition, they are going to produce certain gases such as CO2, such as methane, such as H2S. All of them together you call it as biogas. Works? It's like a monologue. Yeah. Done? Okay. Now over here. Are you? Yes, now we can read. So primary treatment is physical removal. It removes large and small particles from the sewage. Filtration means removes floating debris which is there. Sedimentation removes the grit, that is soil and all, and small pebbles which are there. Okay, the primary sludge is the settled solids which are there, and the effluent moves to the secondary treatment. Whatever effluent is there, it is going to move towards the secondary treatment. Now first thing, aeration process, where the primary effluent is passed into aeration tank. Aerobic bacteria form flocks, that is bacteria plus fungal filaments. Microbes break down organic matter, reducing the biochemical oxygen demand. Lower BOD, that means reduced pollution potential. If there is lower BOD, that means there is less organic matter. If there is less organic matter, obviously the pollution is less. Okay, so lower BOD is equals to reduced pollution potential. Now the next thing is sedimentation process, where the effluent is moved to a settling tank. Okay, bacterial flocks sediment forming the activated sludge. A portion of activated sludge is sent back as inoculum. Anaerobic digestion is the next process. Remaining sludge is sent to anaerobic sludge digester. Anaerobic bacteria digest the sludge, producing what? Biogas such as methane, CO2 and H2S. The final effluent disposal. Treated effluent is released into rivers or the streams, and that's all. I hope you got it. Okay. Children, are you enjoying the class? If you're enjoying, do let me know. Do let me know if I am fast. Well, I'm trying to make it a bit fast, because obviously you might have ended your PU2 examination and all, or probably you might have at least read it for previous time when you were giving board, let's say pre-boards and all these things, that is preparatory exam. So I want you to pace up, because we do not have too much of your time. I do not want you to invest too much of your time into learning one-shot videos, because you have to actually solve questions as well. So do them very, very fast videos and then solve questions. Okay. Now, is there a plan to reduce the river pollution? Yes. So urbanization has increased the sewage production, obviously, because of lots of us living in a city, my dear children. If you see Bangalore itself, we are in Bangalore, and Bangalore is too much urbanized now. There is no place to walk around like that. It's not like that. We do have places, very beautiful places, but still, you see lots of people are in there. Now what will happen? It will increase the sewage production. Now insufficient sewage treatment plants lead to untreated sewage being discharged into rivers. So obviously when the waste accumulates, my dear children, it is going to cause a lot of trouble. People are going to put it in the rivers directly and things like that. This causes pollution and spreads waterborne diseases, which is one major concern, my dear children. So what are the plans that have been put up by the government? There is Ganga Action Plan and this Yamuna Action Plan. It aims to reduce water river pollution. More sewage treatment plants are planned to ensure only treated sewage is discharged. That means when we say we are putting into river and water, make sure that it has underwent what? Sewage treatment. Visiting a sewage treatment plant can be educational and informative. So if you have got a chance, definitely go and visit it. So Ganga Action Plan and Yamuna Action Plan is the plan that we have. Now we have biogas. How is biogas production happening? I already told you. Okay, so biogas is a fuel gas mixture. It contains of methane, CH4, oh sorry, I wrote CH3. Okay, CH4, CO2 and H2. Okay, CH4, CO2 and H2. Please remember, biogas does not just mean methane. It is a combination of gases. This is a very important question. Now, methanogens are there, that is example methanobacterium, that produces methane in anaerobic conditions, my dear children. It is found in sewage sludge and rumen of the cattle. In the rumen of the cattle, my dear children, they also have methanogens in them. In the rumen. Why? Because what happens is that this methanogens which are there, methanobacterium which are there, it is not directly going to break down the cellulose. You know, cows, basically cattle, they eat plant-based products, and plants, they have cell wall, and the cell wall is made up of cellulose. So to break down the cellulose, you have some bacteria in their stomach. Suppose I am a cow, I will be having a certain amount, not stomach, basically digestive system. Okay. So there will be certain bacteria in the digestive system which will be breaking down this particular cellulose, my dear children. So what happens is that methanobacterium are also there. So they are not going to directly break down cellulose, but they are helping in the process. So when the breakdown of cellulose happens, there are so many acids and everything being produced. So to convert them into methane, this plays a very, very, very important role. So we definitely have, not we, basically the cows and the cattle, other cattle, they have methanogens in their rumen. Now humans cannot digest cellulose unlike cattle. I hope you know that. We cannot. That is why we do not eat raw food, because we do not raw plant, because we do not have the things to break down the cellulose. That's why we boil and eat it. Okay. Now so this is a biogas production plant, my dear children. This is a very important thing. Over here there is an area where you put the dung, where you put the cattle dung, and I told you in the rumen of the cattle what do you have? Methanobacterium. So obviously in the dung also you will be having the methanobacterium. Now the methanobacterium or the methanogens which are there, you're going to mix it with water and make it go down to this particular thing, that is a digester. That is what? A digester. Now digester is made sure that it is in anaerobic condition. That means there is no oxygen up here. There is no oxygen up here in the digester, and when there is no oxygen up here in the digester, what will happen? The methanobacterium which is there, it will be acting on the organic matter present over here and releasing biogas. This gas will be given out with the help of an outlet pipe, my dear children, to kitchens or whatever it is needed. Now whatever thing is left behind after releasing the gas and everything, you have certain things left behind, right? So those things can be put down here, and that can be taken as fertilizers as well as manure. That can be used as fertilizers as well as manure. Okay. I hope you got it. Now development and application of biogas. So it was developed by Indian Agricultural Research Institute, IARI, Indian Agricultural Research Institute, and also Khadi and Village Industries Commission, KVIC. Very important question. Okay, you might be asked to basically put the full form, you should be knowing about it. So it is encouraged in village for sustainable agriculture. Visiting a biogas plant would give you a practical learning. Yes. Okay. So Indian Agricultural Research Institute and Khadi and Village Industries Commission is very much important. They are the one who developed the program and everything about biogas and production and things like that. So methanobacterium are there in the rumen of the cattle. Do we have them? No, 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 no. We do not have it. Okay, cattle have it. Okay, and when you talk about biogas, it's a combination of certain gases such as methane, such as CO2, such as H2 and also H2S. Okay, it is not mentioned here, but you should know that. Now, my dear children, the second last part of your chapter is biocontrol agents. How are microbes going to be used as biocontrol agents? First of all, what do you mean by biocontrol agents? For example, we usually use pesticides to kill the pests, right? To kill the pest. Now, what if? And these pesticides are going to increase the chemical. These chemicals are going to seep down to the soil. From the soil it gets into the water. This is the water that we drink and we get affected. You know DDT? DDT was a chemical that was used in Kerala, okay, in Kasaragod district of Kerala. If there's some Malu over here, you might be knowing about it, or else, okay, let me just hear me out, okay. So basically this was used, and what was happening? Slowly the people around that area, wherever DDT was used, they have had certain problems, okay, mental retardation and also deformity which was happening in their body. So later DDT was banned. So there are so many ill effects of what the pesticides that we are using. So obviously if there is some other organism, that can directly eat the pest, so it will be easy. It will be easy, right? So people thought about that and came up with an idea. They saw that there are certain organisms which can be grown up, and these organisms can be eating on the pest, and Basically, in Hindi, there is a a saying, "Dushman ka dushman dushman." So, enemy's enemy is enemy. So it's like that. But we should not be having enmity with anything. For pest, obviously, we cannot make them grow. So we have to kill them. So first thing, my dear children, you should understand what are biocontrol agents. So it relies on natural predators and microbes instead of chemical pesticides. So if the ladybird is okay with eating the aphid, then let it eat. No, anyway, we do not want the aphid. So naturally, it is happening, and it is very good for us. Now, organic farming promotes what? Promotes biodiversity for sustainable and balanced ecosystem. Obviously, this will lead to what? Balanced equation. Sorry, not equation. I went to chemistry. Balanced ecosystem. Now, pests are managed, not eradicated, to maintain the natural food chain. Make sure the pests which are there, it is just managed. It should not be completely eradicated. If it's completely eradicated, then we would not have an ecosystem. If we kill if the ladybird eats all the aphids, then what will the ladybird eat later on? So we have to make sure the food chain is stable. So it's not completely eradicated. Remember that. Now, encourages understanding of, okay, that is not needed. Now, first thing, example of biocontrol agents, you have predatory insects, my dear children. You have two insects. One is ladybird beetle, which is a beetle which has red and black spot on them, and one is dragonfly. So, ladybird beetles are going to feed on aphids, my dear children, are going to feed on aphids which are crop pest. I want you to go to Google. Okay, use your Google to search what is aphid. When you see the aphid, you remember it better. Okay, so ladybird is going to feed on the aphid. Ladybird is a beetle that feeds on aphid, which is a crop pest. Okay, talking about dragonfly. Dragonfly, so you might have seen. It feeds on mosquito larvae, my dear children, helps in what? Mosquito control. Even though it's not a plant pest, because male mosquitoes eat the plant sap and things like that, it feeds the plant sap and things like that, it can be a pest, but again, yeah. So for mosquito control, what do we have? We have dragonfly. So ladybird, beetle and dragonfly, ladybird for aphid, dragonfly for mosquito larvae. Okay, their stomach is full. Now let's talk about microbial biocontrol agents. So these were insects. Now microbes such as bacteria, fungus and everyone. So first one is Bacillus thuringiensis. That's a bacteria. That's a bacterium. I hope you know that. Okay. So this particular bacteria, my dear children, it has certain genes in their genetic material that is known as cry gene. That is known as cry gene. And cry gene is responsible for production of toxins. Cry gene is responsible for production of toxins. But suppose I am the bacteria, in my body these toxins will be in protoxin state. That means they are not active, they are inactive toxin. When will they get active, ma'am? So basically, suppose there is an insect. Okay, there is a butterfly caterpillar, suppose there is a butterfly caterpillar which is eating a leaf, and on this leaf there is a bacteria. So when the leaf has been eaten up, this bacteria is also going to go inside the butterfly caterpillar's body, and it is going to reach the midgut. Which region? Midgut. Okay, it reaches the midgut region. We have studied this in biotechnology and its application. Okay, so it reaches the midgut region, and what happens? This protoxin which is there, it converts into active toxin, my dear children. And you might ask, why? How? So basically, in the midgut of the butterfly caterpillar, there is alkaline pH. What pH? Alkaline, basic pH. And in this basic pH, the protoxin which is there, it becomes activated. And when it becomes activated, it is going to lyse down. It is going to break down the cells there, epithelial cells in the midgut region. And when it does so, basically the whole organism is going to die. Okay, it is going to die. So basically, how do we use this? So bacterium whose spores kill butterfly caterpillars when eaten. So it is available as dried spores. It is available, Bacillus thuringiensis is available as dried spores in sachets and things like that. So it is mixed with water and sprayed on the plants such as brassicas like cabbage, cauliflower, all come under brassicas, and also fruit trees. Now what will happen? In the caterpillar gut, the Bt toxin is released, killing only the caterpillar without harming what? Other insects. Now Bt cotton is genetically modified crop containing. Okay, so apart from putting the spores, my dear children, you can also insert the gene which is there directly into the plant. And when you do so, basically the plant will start releasing the toxin, and when an organism comes and eats it, it will die directly. So either you put it as a spray like spores, okay, otherwise basically you have the toxin genes. You can use recombinant DNA technology to put this gene inside the plant, that is known as Bt cotton and things like that. So for example, in cotton plant, if you are putting the Bt gene, cry gene, it is going to be known as Bt cotton. So that is one way. Okay, now it is for butterfly caterpillar and all the insect caterpillars actually. Now going ahead, we have fungal biocontrol agents. Okay, and fungus also acts like a biocontrol agent. So we have trichoderma fungi, my dear children. Trichoderma polysporum we studied, right? Now trichoderma polysporum was used for production of cyclosporin A, which is an immunosuppressant, just if you forgot. Okay, so now the trichoderma fungi, which is actually living in the roots of the plant. It lives in the roots of the plant. And you know what? They feed on other fungus which is going to act like pathogens. They feed on other fungus which is going to act like pathogens. So free living fungi, it is a free living fungi found in the root ecosystem, effective in controlling the plant pathogen, that is disease controlling microbes. Plant pathogens, what? Plant pathogen? Fungal only. So fungus is going to attack the fungus, okay. Now there is viral biocontrol agents. There are certain viruses also, such as baculoviruses. Such as what? Baculoviruses. Now it belongs to the genus. It belongs to the genus Nucleopolyhedrovirus. Very important information. So this question is usually being asked. So baculoviruses belong to Nucleopolyhedroviruses. It infects only specific insect pests and arthropods. Arthropods as in insects only. Okay, insects and everything comes under arthropods. So arthropods is a big category. In that you have insects, inside you have so many other organisms as well. So it is eco-friendly, does not harm plants, mammals, birds, fish or beneficial insects. It is useful in integrated pest management and in ecologically sensitive areas. So please remember, viral biocontrol agent, that is baculoviruses, belong to the genus Nucleopolyhedroviruses, and they are there for specific insect pests and also arthropods. Okay. The fungal biocontrol agents, you have trichoderma fungi, which is going to feed on other funguses in the root ecosystem itself. Okay. And then you have bacterial, that is Bacillus thuringiensis. You have insects, that is ladybird and dragonfly. What are they feeding on? That is also very much important. So over here aphids, over here mosquito larvae, over here basically the caterpillar, insect caterpillar, butterfly caterpillar, whatever you want. Okay. Larvals, actually insect larvae. It acts on insect larvae. Fungus, that is trichoderma fungi, it acts on the fungal root pathogens, okay. And viral, you have baculoviruses, which infects specific insect pests and also other arthropods. Works? Chalo. Going ahead, we have biofertilizers again. So we use fertilizers. Now these fertilizers can be, obviously we are putting some chemicals, right. So this can change the soil fertility level and things like that. So why don't we just depend on organisms who can increase the amount of nutrients of the soil? Basically, you might have seen that certain bacteria are there that fixes the nitrogen. So basically the soil is getting enough amount of nitrogen. There are certain organisms that gives phosphorus, so the soil is getting enough amount of phosphorus. Get it? So there are different things out there, and these organisms basically who are responsible for enhancing or enriching the soil nutrient quality, they are known as biofertilizers. So they help reduce chemical fertilizers use, preventing what? Environmental pollution, my dear children. You have main sources such as bacteria, fungi and the cyanobacteria. Okay, so cyanobacteria are going to come under bacteria itself, but let's put them into different category. General bacteria and cyanobacteria, we are putting in different category. Okay, so first is nitrogen fixing bacteria. So remember, they can ask you what is a bacteria that fixes this thing, so then you have to put them. What is cyanobacteria, then you have to put them. That's why I made it a separate thing. So nitrogen fixing bacteria, you have two. One is rhizobium and the other one is azospirillum and azotobacter. Azospirillum and azotobacter. So rhizobium, where do you find them? You find in the root nodules in the legume. So in the leguminous plants, such as what do you call, such as what do you call the plant? Oof, I forgot. The green color, beans. In the beans and all, they are legumes, okay. Beans are all legumes. So in the root nodules and the leguminous plants, they are there, okay. The rhizobium is there, and it fixes the atmospheric nitrogen. So atmospheric nitrogen has 3 bonds. N2 has basically 3 triple bonds. So very basic, it is very difficult for the plant to take in like that. So basically this particular nitrogen is fixed. It is broken down in other forms, ammonia and then nitrate and nitrites, and that's how the plants use it later on. So this fixation is done by the rhizobium. Even azotobacter and azospirillum, it's free living bacteria that enriches what? Soil nitrogen content. So both the bacteria for nitrogen, rhizobium and azospirillum and azotobacter. Now you have fungal biofertilizers, my dear children. You have fungal biofertilizers. One is mycorrhizal fungus. So I'll tell you one thing before going ahead. So you have 2 association. One is spelling of, okay, I always tend to forget, mycorrhiza, and the other one is lichen. Listen to me carefully. Mycorrhiza is a combination of plant plus fungi. It is a symbiotic association of plant plus fungi. Lichen is an association of algae and fungus. So basically what happens, both the plant and algae, they are responsible for the they are autotrophs. They can produce their own food. They can produce organic matter with the help of sun and everything out there. So they are going to produce the organic matter and give it to them. That is the fungi. What does the fungi do? Fungi basically can seep into different things and can absorb what? Nutrients. So in the case of mycorrhiza, the fungi is going to take in phosphorus. Take in what? Phosphorus. Please remember the name. Phosphorus is the one that is being taken up by the fungi and gives to the plant. Plant will give organic matter. Similarly, in the case of algae also, algae will give organic matter, fungi will give nutrients. Okay, that is how things work. Okay, so mycorrhizal fungi is there, which is a fungal biofertilizer. Remember again, mycorrhiza, it belongs to the fungus which is there, belongs to the genus Glomus. This is very important. Okay, they might ask you example of mycorrhizal fungus, so it is Glomus, or basically genus, it is Glomus. So it forms a symbiotic relationship with the plant roots. It is found in what? Roots, my dear children. So it absorbs phosphorus from the soil and passes it into the plants. I told you. So basically fungus has hyphae which can get into the soil just like roots and take in the phosphorus which is there. It provides resistance to pathogens, tolerance to salinity and drought, and improves plant growth. Now this is also very much important. There's a question that is being asked, what is a thing that is not provided? Okay, not provided by mycorrhiza. So it provides resistance to pathogens. Pathogen resistivity is also provided. Tolerance to salinity and drought. That means the plants which have mycorrhizal symbiotic association, they are tolerant to salinity. If there is too much amount of salt in the water, salt in the soil, or basically there is drought conditions, still they will be able to survive a bit. Okay. Now it improves the plant growth as well because we are providing nutrients. So these three things are very much important. Now if one more option is added, it increases the yield. It increases the yield. That will be wrong. That is not given any NCERT. So that is a wrong option. Only these things are there. Okay. So mycorrhiza basically provides all this thing. Mycorrhizal combination provides all this thing, nothing else. Please remember it. I am stressing on it. This is why I asked you to take the pen and write it somewhere. It's given in NCERT. You have to underline. You have to make sure you know this. Now fungus benefits by receiving organic nutrients from the plant. I told you, fungus will be receiving but organic nutrients because they are responsible for making the food. Organic matter. I hope this is also clear. Now you have cyanobacteria, there's a blue-green algae, my dear children. Example is Annabana, Nostoc and Oscillatoria. Three of them, they are cyanobacteria. What do? They fix atmospheric nitrogen again. They are also going to fix nitrogen. Okay, and they increase the soil fertility. They are going to act like a biofertilizers in paddy field as well, very much important. And they also add organic matter. So you know, they can basically fix the atmospheric oxygen, and cyanobacteria, blue-green algae, they have the property of doing photosynthesis, my dear children. So since they are doing photosynthesis, they are also going to give organic matter to the soil, increasing the soil fertility. They are going to act like biofertilizers in the paddy fields as well. So please remember all this thing. Again, we will segregate and learn. So first thing, you have nitrogen fixing bacteria, that is Rhizobium as well as Azospirillum and as well as Azotobacter. Okay, all of them, they are going to fix nitrogen. Rhizobium is found in the root nodules of the leguminous plants, okay, or the legumes. Now we have cyanobacteria, they are also fixing nitrogen. What do we have? Annabana, Nostoc and Oscillatoria. So they fix nitrogen, they increase the soil fertility, they act as biofertilizers in the paddy fields, and they add organic matter because they are responsible for doing photosynthesis as well. That is why they are known as blue-green algae. They have the pigments in them. Okay, now we have mycorrhizal fungi. So in the fungi, the genus is Glomus, my dear children. And what does the fungus do? Fungus basically absorbs phosphorus from deep soil and give it to the plant. Okay, and what does the plant do? Gives the organic matter. There are three things given by this combination. One is resistance to pathogens, second is tolerance to salinity and drought, and the third one is improved plant growth. Okay, apart from it, if anything comes, put a strike on that. Cool? Works. And with this, we have completed the whole chapter. Now you tell me, is this chapter difficult anymore to you? So we'll just revise, okay? So microbes in household products which were there, my dear children. There we spoke about lactic acid bacteria, that we spoke about, listen to me carefully, just listen to me, okay? There is baker's yeast, that is Saccharomyces cerevisiae in bread making. There is toddy, that is produced from fermented plant sap. There is cheese production, that is Swiss cheese by Propionibacterium shermani, that produces holes, that is CO2 holes, and Roquefort cheese, which is ripened by a fungus. Then we spoke about the industrial products. So in beverages, we need brewer's yeast, my dear children, that is being used. It produces ethanol, that is alcohol. Okay, and apart from CO2, CO2 is also being produced in fermentation. Now, type of alcoholic drinks, without distillation you have two, that is wine and beer. With distillation you have three, whiskey, brandy and rum. Then we spoke about other acids that are being produced. So citric acid by Aspergillus niger, which is a fungus. Acetic acid by Acetobacter aceti. Butyric acid by Clostridium botulicum, which is again a bacteria. Acetobacter is a bacteria, okay? If I say fungus. Now there is ethanol being produced by what? Saccharomyces cerevisiae, which is a fungus. Apart from that, we studied about certain enzymes being produced such as lipases, okay, which basically removes the oily stains. It is used in the detergents. Apart from that, proteases and pectinases, which is added in the bottled fruit juices, my dear children. And apart from that, we spoke about, okay, one more enzyme is there, that is streptokinase, which is produced by streptococcus. It is going to act like a clot buster. It is going to bust down the clots. Okay, streptokinase produced by streptococcus, which is a bacteria. Now, whenever term coccus and everything come, just understand it's a bacteria. Now we have what? Certain bioactive molecules being produced. One is cyclosporin A produced by Trichoderma polysporum. What does it do? Trichoderma polysporum is a fungus, and cyclosporin A is going to act like an immunosuppressant. Then there is statin, which lowers the blood cholesterol level. It is produced by Monascus purpureus, my dear children. And then we spoke about antibiotics. Basically the first antibiotic was discovered by Alexander Fleming, okay, and it was penicillin. So he was growing staphylococci. Okay, he was growing staphylococci, but then he found that there is a mold, Penicillium notatum, which was releasing penicillin that did not allow the growth of this particular bacteria. So the first antibiotic was discovered. But then Ernst Chain and Howard Florey came in and they expanded things more. And in World War II, basically this was used up. Okay, this was used to treat the soldiers up there, and they got Nobel Prize. The three of them, they got Nobel Prize as well. Now we spoke about then sewage treatment. You have primary and the secondary treatment. Primary treatment as in you're talking about filtration and sedimentation. Secondary treatment as in you're talking about aeration tank. Okay, in aeration tank, if the level of BOD goes down, that means everything is broken down. Okay, and flocks are also being made, remember that. Then it is put to sedimentation tank, settling tank, where the activity settles down, the water is being removed, the activity sludge, little amount is put back to the aeration tank. The rest of the thing is been put into anaerobic sludge digester, where anaerobic bacteria are going to eat on this activated sludge and produce biogas. Biogas includes methane, CH4, CO2 and H2S, okay, or basically H2. Now, two plants, Ganga action plant and amino action plant. Then there is biogas, I told you. And where do you find methanobacterium? Naturally in the rumen of the cattle, because there it helps in the breakdown of cellulose, not directly, indirectly, but they definitely help. Do we have cellulase enzyme? No, we do not have cellulase enzyme. Okay, so basically how is biogas plant working? What do? We add some dung and water. The dung will be having methanobacterium and also organic matter. So this is passed to anaerobic condition, where it is going to act on the organic matter present in there and is going to produce biogas. This biogas is taken out through an outlet pipe to the kitchens and electricity pumps and things like that, and whatever is left behind is used as fertilizers and manure. Cool. Now, okay, it was developed by Indian Agricultural Research Institute, that is IARI, and then Khadi and Village Industries Commission were also behind the development of biogas plants. We have biocontrol agents, my dear children. We have insects first, that is ladybird and dragonfly. Ladybird feeds on aphids, dragonfly feeds on mosquito larvae. Talking about bacterial biocontrol agent, we have Bacillus thuringiensis, which can be put down as spores mixed with water. Apart from that, you can put the gene directly also. What is the name of the gene? Crygene. What does it affect? Basically the insect larvae or the butterfly caterpillar, all these things. Okay, then we have fungal biocontrol agents, that is Trichoderma, which is basically in the root ecosystem. It is going to feed on the fungal pathogens that is there in the root ecosystem only. Then we have viral biocontrol agents such as baculoviruses, which is from the genus Nucleopolyhedroviruses, my dear children, and they feed on or they infect basically certain insect species and also arthropods. Works? Am I missing out anything? No. And then we have biofertilizers, that is nitrogen fixing bacteria, Azospirillum, Azotobacter and Rhizobium. Rhizobium is found in the nodules of the leguminous plants. Okay. And then we have Cyanobacterium, that is Anabaena, Nostoc and Oscillatoria. Three of them, they fix nitrogen only. They are increase the soil fertility. They put down organic matter because they are responsible for photosynthesis as well, and they basically act as biofertilizers in paddy field. Apart from that, we have mycorrhizal fungus. This fungus belongs to the genus Glomus, and this fungus is responsible for capturing phosphorus from deep soil. Okay, and basically whenever mycorrhiza is there, it is going to help in protecting from pathogens, okay, resistance to pathogens, tolerance to salinity and drought, and also improves the plant growth, but does not do anything else. Okay, so please remember that. That's all. Works. So that's all, my dear children. We have completed the chapter. I first of all, I want to thank you all. You did a great job learning this chapter. Kudos to all of you. Please don't be worried. We are going to cover every chapter like this. We have almost covered everything. I don't know how it is published to you guys, but yeah. So I hope you liked the video. If you understood what I'm trying to teach, definitely let me know. If you have any doubts, definitely surely let me know. I always tell, if there are certain questions which you're not able to solve, write down the question. I will solve it for you. Okay, the MCQ questions. So if you're finding it difficult, first of all, try for yourself, and then if you're not understanding or if you're confused, you can ask to me in the comment section. Works, I'll be replying it to you on priority. So that's all my wonderful, wonderful champions. If there is anything else that you want my help with, definitely do let me know. So do not forget to like, to share, to subscribe, and also to comment down. And please do not be worried about any of your exam. Just give maximum test, my dear children. If you have not joined the Abhyas KCET test series, join it. It will be really beneficial for you. And I think that's all. It sums up everything. So bye bye. Take care.